Hi guys, today we are going into gene regulation in eukaryotes and prokaryotes. The first section of this video will be covering about the gene regulation in eukaryote. Gene regulation in eukaryote organisms are much more complex compared to the gene regulation in prokaryotes. There are many different levels in eukaryotes gene regulation, and what we are focusing for today is the level of transcription itself, because the most common method of the regulating of the eukaryotic gene is the altering the rate of transcription of those genes. So we'll be looking into the transcription level itself. What we're seeing in front over here is the basic component of the gene regulation in eukaryotes. There are 5 basic components. There are enhancers, there are intron and exons, and these are introns. And this is a transcription start site, which is called as TSS, and these are the promoters. Okay, let us go in detail with each of the basic component of the gene regulation. These are exons and introns. In a normal sequence of DNA, exons are DNA sequences that encode for a polypeptide, while introns are the sequences that do not code for anything useful, and they eventually get removed through the RNA splicing method, which will eventually left with exons only. So exons are DNA sequences that encode for polypeptide, and introns that do not code for anything useful. And the next component is the transcription start site. Okay, what is the use of transcription start site? This is the site where RNA polymerase II binds to transcription start site and will start the transcription process. RNA polymerase II is a complex of 12 proteins that can synthesize the mRNA. The next component is the promoter. Promoters are divided into two, which are co-promoter and the upstream promoter. We simply call it upstream because we make the transcription start site as our starter, and the left of the transcription start we call it as upstream, and the right of the transcription start we call them downstream. So the upstream promoters are these, and the co-promoters are these. Okay. Co-promoter is a sequence of DNA located 40 base. So the difference over here is the 40 base upstream of the start site. It is found in all eukaryotic genes. Why does it found in all eukaryotic genes? Okay, there's a reason why it's found in all eukaryotic genes. Because the co-promoter remain consistent throughout all the eukaryotic genes. Remain consistent means that it remains as Tata box, TA and TA. What does this means? That means that it is TATA, the sequence of nucleotide TATA. Okay, and the followed by the upstream promoters. Okay, so the upstream promoters. Okay, let us go into detail with the Tata box. Over 50 proteins can bind to the co-promoter, forming an important complex needed for gene expression. This complex includes the transcription factor 2D, transcription factor 2B. Okay, this transcription factor 2D consists of Tata binding protein, which is TBP, which recognizes the Tata box and attached to it, while transcription factor 2b helps the TB protein to interact with the RNA polymerase which is present in transcription start site and to start the transcription level. Okay, the next component is the upstream promoters. Okay, upstream promoters are the segment of DNA found upstream of the core promoter. So these are the core promoters. So upstream of the core promoters are the upstream promoters. Okay, they can also bind to activator or repressor protein needed to regulate the gene expression. So it's either you activate the gene or you block the gene, depends on the upstream promoters. Okay, and the last component is the enhancers. Okay, these are the DNA segments typically located far away from the gene to gene. They can be either upstream or downstream. These enhancers binds to the specific transcription factors that increase the rate of transcription. Okay, let me go into that in detail. Okay, as y'all can see, this is the DNA sequence, right? Okay, these are the enhancers. Okay, what does the enhancer do is it combine with the transcription factors TF and it will bind itself to the transcription start site where the RNA polymerase is present and to the Tata box over here, Tata box, to begin the transcription level. Okay, this will eventually increase the rate of transcription. Once the transcription factor binds on the protein onto the enhancer can loop around onto the promoter region, thereby stimulating transcription of that gene. Okay, that's all for the gene regulation in eukaryotes. Okay, let us go into detail on gene regulation in prokaryotes. So, gene regulation in prokaryote is much more simple compared to eukaryotic gene regulation. So, to understand this, you need to know some basic features on the gene structure. Okay, special DNA binding proteins called repressors are activators. These activators and repressors can be controlled allosterically. The model is operon, and where it consists of two sections of regulatory and coding sections. Okay, we want to control the expression of certain genes. Okay, for example, this is a double-stranded DNA sequence. Okay, from the double-stranded DNA sequence, we would like to synthesize mRNA. From mRNA, we would get the protein. The protein is the ultimate thing. Why is the gene regulation has been happening? So this is simply means that gene regulation is the process of regulating the expression of certain gene that will finally make a protein. Okay, in this structure, what we can see is the promoter, operator, structural genes, and the regulatory gene. Okay. There are multiple, as you all can see here, is a structural genes, right? Okay, those are the multiple genes are clustered one after another. All of those genes are expressed by the control of regulation of one promoter only. So this one promoter and one operator control this multiple of genes are called operon. They also control this segment whether to transcribe these DNA sequence into an mRNA or not. That means all the genes are then turned on and off simultaneously simply because of one promoter called as operon. So the process called on off. This simply means that the promoter, the ultimate thing, controls whether to transcribe the DNA sequences into the next level or not. There are intron and exons.